Our topic is antigen-antibody interaction. Antigen-antibody interaction. So, when an antigen come in contact with antibody, okay, there is a reaction takes place. So, that is the basis for, that is basic for humoral or antibody mediated interaction. So, there are three stages in antigen-antibody interaction. The first one is formation of antigen-antibody complex. So, first the reaction, the first reaction is antigen-antibody complex formation. And second one, that is the visible events are taking place when the antigen and antibody are interacting. The reactions are precipitation, agglutalization, etc. Like such of the visible reactions are taking place. The third one is destruction of antigen or its neutralization. So what are the three stages when an antigen and antibody interact means? The first stage is antigen-antibody complex formation. The second one is the visible events like precipitation, agglutalization has to be takes place. And the third one is destroying the antigen or neutralizing the antigen. Binding force of antigen-antibody reaction. So there should be an force in between the antigen and antibody. So that we can see in three factors. First one is closeness between antigen and antibody. If the closeness is more, okay, that is antigen and antibody, there should be a closeness. And the non-covalent or intermolecular forces should be there. And the affinity of antibody. So these three factors determine the binding force of antigen-antibody reaction. When an antigen and antibody are closely fitted, so they are coming in very close and they are fixed very closely, so then their binding site will be great. When they are apart from each other, the binding strength will be low. So the bonds between the antigen and antibody the bonds between the antigen and antibody are non-covalent in nature. So there is some other forces and there is some other bonds also. In not only the non-covalent bonds, here the hydrogen bonds, electrostatic bonds, van der Waals forces and hydrophobic bonds are involved in this, antigen-antibody binding site. So the antibody affinity, that is the strength of the reaction between a single antigen determinant and a single combining site of the antibody. So there should be an affinity to bind with the antigen and antibody. So a single specific antigen has to be find out first. After that, that particular specific antigen should be binded with the antibody site. Okay. The chief application of antigen-antibody reactions are to determine the blood group for transfusion. So you have to know when the blood has to be transfused, first you must know the what blood, as we have discussed about the A positive, that is the A blood group has B antigen. If you allow the B group blood in A group, the person will be die. So that means you have to check the blood groups before transfusion. To study the serological assignment of exposure to the infectious agents. So the infectious agents, what are the serology you are exposing, that you have to be know first. You have to assess it and what are the infectious agents, that is infectious agents, this serological should be exposed. Okay. To develop immunoassay for the quantification of various substances. So you have to create the immune assay. So because the immune assay has to be stand, it has to be there to overcome the some other substances. It has to fight. Okay. To detect the presence or absence of proteins in the serum. So it is used to detect the presence or absence of protein in the serum to determine the characteristics of certain immunodeficiency disease. So these are the chief applications. So first you have to determine the blood group of transfusion. Next one is the serological assessment of exposure to the infectious agents, to develop immunoassay for quantification of various substances, and to detect the presence or absence of proteins in serum to determine the characteristics of certain immunodeficiency disease. So these are the chief application of antigen-antibody reactions. Different types of antigen and antibody reactions. So when a soluble antigen and antibody, so if they are coming in contact, what will happen? You can see a visible precipitate formation, and this reaction is called precipitin reaction. That is, a soluble antigen binds with the antibody, while that time you can see a visible precipitate. That is called precipitin reaction. The antibody which produce precipitate around the antigen, that is called precipitants. So when a particular, that is particulate antigen interacts with its antibody, so the particular antigen interacts with the particular antibody, it forms a clumps around it, or agglutinin takes place. That reaction is called agglutinin reaction. So the antibody bringing agglutinin reaction, it is called agglutinin. So here you can see the that is antigen antibody interaction. Okay, so first one is neutralization. So first one we will see here. So first one, it is neutralization of, that is, exotoxin toxoid. So this is the bacteria, and you can see the virus and the bacteria. This one is the bacteria and this one is the virus. How the antibodies and the antigen are binding, okay, through some force. Like this stage, the neutralization takes place. And agglutinization, so agglutinization, the bacteria, and it got clumps means it is in a clump. So this particular antigen got agglutinized. Next, precipitation, you can see the precipitated molecule, antigen molecules here. So soluble antigen, so they form the precipitants, that is the antibodies. Here it is called as precipitin, and the reaction is precipitation. So this is so this one is the complement system. You can see the complement system, and you can see the foreign cell, that is the foreign cell, and you can see how it enter. So it is hiding and the complement molecule here, okay. And after this, it leads to the cell lysis. After the what are the stages going to take place? The antigen, if enters into it, so after that what it happens means, you can see the cell lysis here in this. In this you can see the cell lysis takes place here with the antigen, the antibody and the antigen binding it, and the cell lysis means cell lysis will be taken place. And you can see the inflammation portion here, that is also one of the reaction of antigen antibody reaction. And here it is phagocytosis, that is the cell eating. It eats the antigen, that is the bacteria or virus which in. So it eats that is the immune system cells. The immune cells will eat the antigen. Opsonization. So opsonization, it is a process of pathogen which is marked by injection and destruction by the phagocytosis. So opsonization, so pathogen is find out. So here you can see, no. So in this here you can see the pathogen. So the pathogen was eaten by the immunocells. So okay, like macrophages, it got engulfed through the phagocyte. So opsonization here, the binding of opsin, that is the antibody to a receptor on the pathogen cell membrane. So this pathogen enters into the phagocytic cell, it creates a opsin, okay, opsin, okay. So opsonization, that is, it is a process in which the pathogens are coated with a substance called opposin. So here the opposin is the substance which covers the pathogen. And due to the covering of the pathogen, it got marked. The pathogen was got marked and it was digested by the phagocytic cells. So, and it was caused due to the destruction, that is immunocytes. That is the immune system works out on the pathogen and destroys the pathogens. So, it is an efficient phagocytic method, opsonization. Next, neutralization. So, this is also one of the reaction of antigen antibody, and here the harmful effects has to be eliminated. Mostly the bacterial exotoxins are, so that here you can see, no, the bacterial exotoxins are, and viral particle, that is the virus by which was identified by the specific antibodies, and it was neutralized with the help of antitoxins, that is bacterial exotoxins or virus has to be neutralized with the help of antitoxin created by the antibodies. So was produced in the host cell in response to the bacterial exotoxin. So responding to the toxoid, which activating, that is this antitoxin is inhibiting the exotoxin or inactivating the toxin. Hope you understand the topic. If you have any doubts, post your doubts in the comment box. I had given the Tamil link in description box. You can go and watch it. If you like the topic, give a thumbs up, share and subscribe to Science Easy Tech channel.